What is the cell cycle? The cell cycle, or cell division cycle, is the series of events that take place in a cell leading to its division and duplication of its DNA to produce two daughter cells.
The cell cycle also includes checkpoints to ensure that cells are developing properly. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted. The cell attempts to either complete DNA replication or repair the damaged DNA. The checkpoints include G1, G2, and M.
The cell cycle includes a growth stage, mitosis, and cytokinesis. Interphase can be broken down into three phases: G1, S, and G2. In the G1 phase, the cell goes through growth. This is considered the first phase of the cell cycle, and during this time, the cell is making mRNA and proteins that it needs to undergo DNA synthesis.
The G0 phase, or resting phase, is a period in the cell cycle in which cells exist in a quiescent state. The G0 phase is viewed as either an extended G1 phase, where the cell is neither dividing nor preparing to divide, or a distinct quiescent stage that occurs outside the cell cycle.
In the S phase, the DNA replicates, creating two copies of each chromosome. The S phase is the second phase of the cell cycle and the most important phase of interphase. Here, the cell is going to replicate all of its chromosomes in order to make a second set for the daughter cell to have.
In the G2 phase, the cell continues to prepare for mitosis and cell division. G2 is the last phase of the interphase, and basically the cell is checking to see that the duplicated chromosomes have no errors.
In mitosis, there are four stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes condense and become shorter and thicker. Each copy of a chromosome is called a sister chromatid. Then, the spindle fibers begin to form as microtubules grow out of the centrioles and move to opposite sides of the cell.
During metaphase, the double-stranded chromosomes move to the center of the cell. Now the spindle is fully formed and the microtubules attached to each sister chromatid.
Anaphase starts when the sister chromatids of each chromosome begin to separate. The centromere that holds the sister chromatids together splits, and then the chromosomes move away from each other. During telophase, the chromosomes reach their opposite sides of the cell, and this is when the new nuclear envelopes begin to form around each group of chromosomes. The chromosomes uncoil and then the spindle disappears. Then the cytoplasm divides. This is called cytokinesis. The formation of two genetically identical cells is the product of cytokinesis and mitosis. During interphase, a cell goes through G1, S, and G2 to grow, and that way it can divide. In mitosis, there are four phases: prophase, metaphase, anaphase, and telophase. In cytokinesis, the cell's cytoplasm divides to create genetically identical daughter cells.